first of all, good morning, everyone. I'm going to start because, as you can see here, we have a very tight agenda, so I don't want that we start later in the beginning. So, well, this session is going to be recorded, so those who are not here and they want to know more about the pilots, they will have the chance to hear the audio or the video after the seminar. So yeah, like we have a tight agenda, as I commented. I'm not going to do like a long introduction. It's going to be just a couple of minutes. I just want to emphasize because, well, we shouldn't expect much progress since what we presented in the annual meeting that was like 2 months ago. But the goal in this seminar is different. In the annual meeting, we emphasize, or the main goal was to prove the link between each pilot and the different working packages and the synergy among the pilots. And also we had like 5-10 minutes to explain everything that we have been done in this year. But now, apart that we have more time, but we have like 20 minutes to explain our pilot, so we can explain a little bit more in detail what we are doing. The main objective of this seminar is to show how each pilot fit into the framework of the OBSGESSION project, how the pilots align with the mission of OBSGESSION. So at the end of these seminars will be clear why do we need those pilots in this project. So I would like to emphasize that please each pilot need to emphasize first of all the novelty, what we are doing differently or what we are doing new, but also why do we need this specific pilot in OBSGESSION, how this pilot aligns with the mission of OBSGESSION and how this fit into this project. So this is the main goal for this seminar. So I'm not going to explain much about how this working package is about because you already know it. You know that we have like 6 pilots. So in the agenda, we are going to explain 3 of them. There will be time for questions. There will be a break after that, and then the other 3 pilots will go on with the presentation and some questions at the end. So please, I want to say that keep the time and I don't go beyond these 20 minutes that you have per pilot. So we should avoid to have a lot of delays at the end of this seminar. And with that, I would like to give the floor to the first pilot. And well, before that, I would like to comment if you can use your laptop and your presentation to share during this seminar, so you will have fully control of this presentation. But I would like to ask you if at the end of the seminar you can put all this material on Teams so everyone will have access to those presentations after. With that, I will stop talking, so I will give the floor to Maria to present the pilot one if there is not any question for me.
Yeah, do we want to switch and take the second pilot first?
Yeah, so maybe. Yeah, I know that you have to leave soon, so I hope that there is still time for you to do the presentation after. So maybe we can move to pilot 2, if that's it okay with Sander.
No problem. I will share the presentation. You can all see the presentation,
yes.
I hope?
Yes, we can.
Okay, perfect. So I will shortly explain what we are doing concerning the progress in Pilot 5.2 on mapping and monitoring of European habitats. And as you can see, mapping habitats is the cornerstone of the EU's biodiversity strategy for 2030 and the new nature restoration law. So it's very important to understand the habitat extent, the conditions and changes that are taking place in those habitats. And our first aim in the pilot is to improve our existing habitat classification methods. So see where we can make improvements, and that's not only on the classification algorithms, that can also be on the input and the training data. And our second aim is to focus more on the change detection, and for that we will both exploit also the Copernicus land cover change products, or the Copernicus land cover products, and we will look also at NDVI time series to look at more at the gradual changes in the habitats. This is some of the progress that we have made. It's a quite long list. We have been experimenting with deep learning classification algorithms for the Netherlands. We have been working, for example, with the Deep Lab version 3 in Jupyter Notebook, and more accurate data sets, because for the Netherlands, for example, we have much more accurate data sets than on a European scale, so it's interesting to look what that does for the habitat classification. CNRS is working on the improvement of the European habitat map, pushing for better model performance. We have been working also on the anomaly detection of the training data, because also in the huge amount of training data that we have, there are still a lot of errors, especially in the accuracy of the location, and this has an impact on the classification results. So, for example, much better integration with land cover data can be, for example, with national land cover data, can be used to identify errors in the training data. We have been working, but that's more related to task 2.2. But important for our task here is the development of a Yunus proxy distribution viewer. This enables us to locate more Yunus habitat locations, for which we can use for training. We have been experimenting with data augmentation to increase the training data for the Netherlands. I will show you also some slides on that. We have been working on the OpenEO, but it works slow and only for small areas, so we will show some slides on that. We have had a meeting with Suka for collaboration on Northern Lapland, especially to assess the European habitat map. We have been working on we will be working soon on the integration masking of the existing European habitat map with the Copernicus high resolution layers, especially to remove non-habitat areas, because still the European habitat maps overestimate the amount of habitats, and if we want to monitor, we have to be sure that we are dealing with the correct habitats. We made also a start with the translation of land cover flows into pressures, because that's also very important to see what kind of pressures we are dealing with, not only those that are removing the habitats, but also in the surrounding landscape, for example, that is causing more fragmentation of the habitats. And we start with, we will start soon with analyzing Copernicus land cover changes. We are also thinking of the Copernicus backbone to use that with regard to the European habitat map to identify changes. So this is the current status of the Yunus habitat map. That has been made already before, also in the ESA project EO for Biodiversity, but still there's an idea to improve this map in different ways, like I mentioned before. These are some of the ideas of CNRS to work on the, let's say, on the, further pushing for better model performance. So also there we can do a lot, also on the data augmentation. There are a lot of opportunities to also in the input data to improve the model performance. Then Then for the Dutch case, I will hand over a little bit to my colleague, De Mantas, who has been doing a lot of the work on this.
I can join you.
You sit on the other side. Yeah.
Yeah, hey everyone. Yeah, so I was working on the Dutch case with deep learning and looking into the 10-meter resolution habitat mapping. Yeah, so currently what we're working with is the data that we have is quite sparse, the labeled UNIS habitat classes. We have a lot of data, of course, because it's 10-meter resolution, but only very few labels. Only 0.003% of the data is labeled. So that is a very good recipe for overfitting. And we really have to look into how to prevent that. And we're actively looking at different options currently. Yeah, so let's go to the next slide, maybe. So currently we have a habitat map ready, where we use the DeepLab V3 model, and the accuracy seems to be quite reasonable. However, there are a few questions about how to really evaluate how accurate habitat mapping approaches are. In terms of splitting the training sets and test sets and whatnot. So we are actively looking into these kinds of questions and preventing overfitting, as I mentioned. To the next slide.
Yeah, it should be. Yeah.
Yeah. Do you have something to say for comparison about with the previous?
No, I think it's just showing some on the top, the let's say the different models that we implied, but also, for example, if we look very much in detail for the European habitat map, using 56,000 national training validation points, still there's a lot of room for improvements, and let's say that the DeepLab is overall a better accuracy, but still there's a lot of room for improvements. And here at least you see the difference between on the right side you have the bottom right side you have the detail, very small detail of the European habitat map, which is, I would say, really good in overall patterns, but that's at 100-meter resolution. While if you work at the 10-meter resolution, you see already that you can see much better the detailed patterns in the landscape, also in this case related to heathland and inland sand dunes. But also there's still much room for improvements, because not all the labeling is done well. But overall you can see that you could say that the patterns are quite similar between the left figure and the right figure. So I would say we are on the right track, but there's room for improvement. This is another example of the inland sand dunes, and you can really see, like on the top figure, which is a Sentinel-2 multispectral image of one date, that you see the detailed patterns at the national land cover classification at 10-meter resolution is better than at the 100-meter resolution, because the inland sand dunes are underestimated and the forest is overestimated. This is the I will not put much detail on say about it, but we developed this UNIS proxy distribution viewer, so you can allocate or find new potential locations for training on UNIS habitats, exploiting the GBIF data and individual species information from the European Vegetation Archive. But the whole analysis is based on 52.2 million georeference plant species records. Maybe still, Damanta, something on the model training and augmentation experiments?
Yeah, so this is what we are currently actively working on. So recently we did some analysis on model training with kind of different scenarios. So we chose to compare training a model with all of the input data that we have. So that is a lot of continuous numeric values and a lot of categorical inputs, such as land use, Copernicus, land cover, and so on. And also compare that with only using a certain small subset of inputs that are relatively easy to obtain. So things like canopy height, elevation, distance to water, distance to sea, and also sentinel imagery. So this one we call numeric only inputs or continuous only inputs. And in addition to that, we also experimented a bit with label data augmentation. So expanding the set of labeled pixels that we're working with. And I can show you a couple of examples. So this is the data that we are originally working with. So these are the available UNIS class labels. And you see that they are quite sparse. Out of the entire image, we only have a few labeled pixels. So that is quite difficult to train a model on. So what we looked into is how to expand it. And we decided to add labels to pixels that have the same land use within a certain radius. So say if we have a labeled pixel on a forest pixel, we add more labeled pixels within the same forest range. So you can see how that looks like in the next slide. And if I switch back and forth, you see that each pixel gets expanded by a number of pixels around itself. And that seems to still contain the habitat patterns quite well in terms of not adding too much noise or potentially faulty labels. So these are two cases that we worked with. And then you can see the model training results here in this slide. On the left, you see some available labels that we trained the model on. And on the right, we see a comparison of the model results with respect to the class probability for a certain class. So in this case, it's a coniferous plantation of non-native trees. And you see there is a difference between non-augmented and augmented approaches, especially with respect to the class confidence. So you see with non-augmented data, the model becomes very confident in itself in terms of, so you have very rough labels, very rough edges on labels. And with augmented data, where you expand the available labeled pixels, the model confidence drops, but it becomes a bit more smooth. And this is a bit of a dilemma to decide which result we really want to have in the end. In the end, I think the augmented result seems to be more realistic in the sense that it's a smooth transition between different habitats. But as you can see in another slide, we also found that the non-augmented approach is quantitatively performing better. So it's predicting the actual labels better, but at the same time, it's too confident in those predictions in our judgment. So it's a bit of a dilemma that we have to discuss about and sort of find solutions to. Yeah, this is currently the analysis that we are conducting. Yeah, so to conclude from these results is that, yeah, augmenting the available data really affects the model behavior that we end up with, and especially with respect to how confident the model is in its predictions. And we have to decide on what sort of confidence behavior we want from the model. Now, the non-augmented model performs better quantitatively, but as I said, it seems overconfident. And also, of course, it's maybe not surprising that the model performance varies quite significantly between different habitat classes. So some habitat classes are predicted very well, especially the more prominent ones, the more distinct ones, like non-native forest plantations. But some other classes, like less prominent ones, are predicted less well. So these are the conclusions. And one possibly interesting idea that we maybe would like to experiment with further is to look into co-occurrence of different habitat types within the same kind of land use, for example, or within the same kind of canopy height level, to judge the confusion between different classes. So some class confusions might not be as bad. So say confusion between different forest classes is not as bad as confusion between a forest and a dune class, for example. So we would have to take this into account, that there are these differences between interactions between different classes. And we'll see how we can best implement this in either the model training or the model evaluation that we're conducting. Yeah, so that's
yeah, and then Stan is taking over a little bit. And Marga, you keep the time. You keep the track of the time that we have.
I think we're running out a little bit, maybe, but I will keep it short.
You can keep it short.
Yeah, so we did a first experiment with the OpenEO platform, with Marcel also showed in Twente. And we tried to download some data for the Netherlands, and we started in Wageningen, where we wanted to have the NDVI. So we did that for, per season for 2022. So you have 4 time images of that area, and it took 6 minutes to process. So that was still good that it worked, and good to see. Then we tried to do the full time series of that year. So basically every time point there's available in a decade. So 10 days. And we got, as you see on the left image, the different images and the NDVI values of the mean of that pic image. And then it took 45 minutes to process only the part of Wageningen, which is a little bit longer than maybe expected. So this is then the result of what we got. You see also the cloud cover, which is removed from the image. So that works fine. So we have some discussion points on that. So the notebooks were good. I think we can also adjust them quite well. And there's an output we get which can easily be opened in GIS environments. However, for a small area, acquiring only 1 year of data, and then each decade, I don't know the English word, but took 45 minutes. So if you want to download larger images or areas, we have to see how that goes. Maybe only not download the image, but only go for the statistics. And the credit limit and how much can be downloaded at once is also a question we still have for Vito, because bigger areas around Wageningen to process in one at once, that was not allowed. So maybe we need to discuss and see what are the options there. And we want to make a comparison with Green Monitor, which we have here. where we can also extract the NDVI time series for the Netherlands and see the difference between the OpenEO and those NDVI values. Yeah, I think that's for now the most important things about OpenEO. So
yeah, thank you, Sam. And then to continue on, let's say, improvement of taking out non-existing habitats from the European habitat map. That's where we will be working on the coming weeks. There's a lot of Copernicus new high-resolution layers available, also for croplands, so that's interesting to have a look at, but also at the imperviousness urban areas. We can remove, hopefully, part of the intensive grasslands. Maybe there's also information on the mowing intensity that can help, because let's say a lot of the grasslands do not belong to habitat. So it will be interesting to make advantage of those Copernicus high resolution layers. And we can also exploit Copernicus data for identifying land cover changes. What is recently available, and we will be working on that now, is the Copernicus plus backbone for 2021. It's at a 10-meter resolution, so very fine information. And we can compare the 2018 with the 2021. The European habitat map is related to data from Corine data from 2018 and a lot of remote sensing data from the period 2018-2020. So it's interesting to see already what has changed and if some of the habitats have been removed, diminished, or there are changes taking place on the surrounding. But there is also the Corine original, the traditional Corine land cover change products available for 2012 and 2018. And we ask now also for the CLC Plus LULU-CF products, which might be interested. We got the data yesterday for 2018, 2021 and 2023. So we have to see if this is interesting data. Another thing is, oh, I see you cannot see it here. But, huh?
Press again.
No, it's not coming up. But we're working also on the land cover flow. So when it moves, let's say from forest to urban, then we know there's urbanization taking place, and this is kind of pressure on the habitats. Even if it's not occurring in the habitat itself, then it's a destruction. But if it's occurring in the surrounding of the habitat, urbanization is pressure on that habitat. So you can really convert the land cover flows into pressures. Yeah, this is the overall draft. So we're working on the mapping and on the monitoring. Also, we want to exploit, seems that this is, I think I have used another version, sorry for that. But also with Vito, it's still on the discussion, the habitat mapping in OpenEO and the change detection. So that's it for now.
Only Finland. So
Finland. I have to.
Wait, I can share my screen.
Yeah, you can show your
2 more slides.
Yeah. I will. Yeah.
Can you see?
So.
Yeah.
Yeah. So we are also working on. We had a meeting with Finland on the habitat mapping in northern Finland. They made a very nice habitat classification or vegetation map. It was based on Earth observation data, but also airborne data, LiDAR data, aerial photos. They did a lot of field work for that, and they exploited machine learning tools to make this map. And it will be interesting, next slide, to show, to assess the European habitat map. How good it is. You see already that it has similar patterns, but we have to look also into the right translation of, let's say, the Finnish vegetation types into the and habitat types into the UNIS habitat type. So to make a good translation is important. Key findings of the from their side is already that 20% of the mountain heaths in the project are in excellent condition, but 80% of them have declined in varying degrees due to heavy summer grazing of reindeers, and it's also effects on the mountain birds and lichens. And there's mass occurrence of Mitari parahoset. I don't know how to pronounce it, but there's also problems with climate change, melting of the balsam mires. So restoration is related to changes in reindeer herding practices, and maybe it will be also interesting to see if we can, with a time series from Sentinel-2, if we can see the decline in the quality of those mountain heaths. So that's it for the moment. Say thank you.
Thank you very much. So we should move to pilot one because I know that Maria has to leave and we are a little bit behind the schedule already. So Maria, the floor is yours.
Yeah, do you mind sharing the?
Yeah, Heidi is going to share the screen.
Yeah, I will share the PPT.
Yeah,
if you
thanks
just
very
give
much.
me a second, I will come. The presenting mode.
So I'll be very short because I'm actually teaching a graduate school seminar. So I just a little bit of time, but I know that Joachim is here. He'll probably be able to answer questions if there is needed later on.
I think this is a wrong one which I shared.
Yeah, that's not ours.
No, sorry.
But
yeah.
So pilot one is about multi-scale, multimodal assessments of biodiversity. And this is led between.
Are you able to see my screen now?
No.
This is not.
We see the wrong one.
Yeah, I'm not sure because I even don't have this document open. Wait, sorry. It's annoying. Okay. Yeah, now?
Yeah.
Okay.
So if you go to pilot one, please. Okay, I might be able to.
So this one?
Yeah, thanks.
Okay.
So the next slide, please.
Yeah.
So next slide, please. So our main goal is to build AI-based models to predict in space and time the response of certain functional and trophical groups of biodiversity across a gradient of alpine environments. Next slide. So what are the locations and ecosystems that we're focusing? Well, I already mentioned forests, grasslands, and wetlands in alpine ecosystems. And we had proposed 4 sites for which we are going to collect both in situ and airborne spectroscopy data. So the Swiss National Park, locations in the French Alps, Legern Forest in Switzerland, and Greifensee for the freshwater environments. Can we go to the next slide, please? So, because Marga asked us to link this to the overall OBSGESSION objectives, so the biodiversity products that we're interested in are related to functional and trophic diversity and species and specific groups of species, but also looking into ecosystem heterogeneity and primary productivity. Can we get to the next slide, please? So our goals, in a very direct way, is to look into the links and examine the potential links between imaging spectroscopy data and maybe LiDAR products and in situ biodiversity data collected at these specific locations. What we want to examine more specifically in terms of the scientific questions are some questions related to how do the changes in the spectral signatures explain multi-trophic compositions, what kind of relations and understanding of biodiversity composition and structure and processes can we have with these airborne campaigns, and in a way is to prepare for upcoming missions such as CHIME and SPG, which will be delivering much higher resolution and spectral and spatial domains for biodiversity assessments. Next slide, please. So what we have coming into OBSGESSION, we had already several studies conducted in many of these study areas that we've proposed. So in France, and Joachim, please complete if this is not complete. So there's plant relevés data, soil environmental DNA metabarcoding, passive acoustic recorders, camera traps data. And in Switzerland, we also have in situ biodiversity assessments, also genetic diversity, especially in Leghorn of Fagus sylvestris, imaging spectroscopy data over time series with APEX and AVRIS airborne campaigns, and also we have flux data from EC towers. Next slide. You've seen this slide before at the ACM meeting that Joachim presented for the French sites. There is quite a bit of existing data, and this will be the foundation from the in situ point of view to develop these AI-based models that link the in situ data to the imaging spectroscopy data. So this will give us different taxonomic resolutions, different taxonomic groups, different methodologies, and we'll see how can we actually use these multi-scale data sets and to harmonize them. Next. So you can already go to the next one. So in 2024, we did the first flight campaigns with AVRIS-4, which is an airborne imaging spectrometer that is now at the University of Zurich. It was built in cooperation with JPL as a sequence of the AVRIS Classic AVRIS NG, and now there's AVRIS 3, 4, and upcoming will be AVRIS 5. How does this look like? So it's an airborne sensor. Here just the representation of what the image data cube might look like, and then we get these spectral response functions that are very detailed from multiple absorption features. Next slide. So we got the data last summer, and we had, well, for the alpine environments, we have a lot of different potential acquisition issues, and so we had geometric correction issues, and so we now have a relatively high spatial accuracy in our acquisition. So this is the data that I received last week. So I think that now the geometric correction is becoming very, very good, which is very fundamental to be able to link it to the in situ data. So this is the most recent data over the Swiss National Park, and in the next slide, please. And this is over Leghorn Forest, the other site in Switzerland. We also had a campaign in September of last year, and in the bottom you see here how the spectral reflectance functions actually look like. So on the left are the original spectra, and then with a little bit of smoothing on the right, you see how accurate the data might look like over different types of targets in the Earth's surface. Next slide, please. Concurrently with the data acquisition from the airborne campaign, we conducted in situ spectral and trait data in Swiss National Park in Leghorn, and we used a sampling structure so that we could link it directly the in situ to the airborne data. So we are currently processing the collection of the samples in the lab for multiple constituents and their composition, and then we aim to link this to the spectroscopy data. Next slide. So it's just for your information of what is the density of the sampling for both the forest and the grasslands. Next slide, please. And now moving on into the plans for 2025. So we're very busy planning the flight campaigns and the in situ data acquisitions. So Joachim, please pitch in if I'm incorrectly describing your CHAMP. So the idea is to repeat spatial representation scheme that has been implemented in the French Alps, also in the Swiss National Park, in other areas of the alpine environments in the country. And so this is basically aimed to cover a gradient of alpine environments so that we can collect different information on trophic structures across this gradient. These are called the ORCHAMP plots and ORCHAMP sampling design. And the idea is that this year we're going to implement this for the in-situ collection. And then we will do imaging spectroscopy over these ORCHAMP gradients to really try to see how biodiversity varies across gradients. Next slide. So the plan for 2025, we will have the Swiss National Parks, and this will be also where our annual consortium meeting next year will take place. So thanks for the votes already. So we will be going to Zernet, that's the majority. Next slide. And we are also examining with the CNRS what would be one of the two locations in the French Alps that we're going to also have imaging acquisition for this summer. Next slide. What we are planning is to collect in situ spectral and trait data as well with the handheld field spectrometer and the trait data that will be used to inform the algorithms. We also are harmonizing our protocols with protocols that are used for SVG and CHIME for the upcoming data products that they will produce as part of their analysis ready products. And we will also conduct in situ measurements on biodiversity data of different kinds. So you see here soil DNA, soundscapes, camera traps, and plant relevaes. Next slide. So how does this pilot link back to the OBSGESSION goals? So we have 3 OBSGESSION goals: harmonized biodiversity monitoring. I think that it's quite self-explanatory, but we will experiment with much different types of ways of collecting biodiversity data and see how can we scale it up using the remote sensing observations. Of course, this gets to the improved use of Earth observation data. So we have already some biodiversity products that we're interested in and that we're developing with Sentinel-2, but we want to use the airborne data to also create this seamless transition to upcoming missions data. And then, of course, knowledge of drivers of ecosystem change. Once we have these models that link biodiversity and may be able to predict its change, then we'll be able to incorporate some of these work together with work package 3 on the detection and attribution framework. So with that, next slide, I think it's the last one. So thank you very much for your attention. And I will have to leave soon, but if there's any questions that Joachim cannot answer, please feel free to drop me an email.
Yeah, thanks, Maria. I'm not sure Andrew was, I think he was trying to ask a question, but I cannot see his hand is raised anymore. So maybe we can proceed to the, yeah, Andrew.
Sorry, yeah, hey, good morning, everyone.
Yeah.
I know I had a question about one of the earlier, but I guess we'll take that in one
Okay.
in one hit after all the presentation. Yeah.
Okay, thanks, Andrew. Thanks. Yeah. Maybe we can proceed with the third pilot 3.
Yes.
Rashid, if you are ready. Yeah.
That would be me, right? Or is it? Or are you?
I will present my part for downscaling.
Yeah.
Then, if you have something to add, then you can present. So, yeah.
I have some very. So, shall I just show a couple of broad overview slides to provide the context, and then?
Okay, that's good. Yes.
You can follow with the detailed workflows. Yeah. Sorry, we didn't coordinate this very well between us, but we're into different time zones. Give me a second.
Sorry, Ben, just a quick question, Rashid.
Yeah.
So are you going to present your part from your side, or you want me to continue like what I did for Maria?
I will present my part, but Ben will start.
Yeah, I know, Ben, he wants to present. Yeah, thanks.
Yeah.
Clear.
Yeah, so sorry for the confusion. Just give me a sec to get organized technically here. You can see that, I trust? Yeah, so pilot 3 is the full title from the original proposal is Monitoring and Forecasting Forest Ecosystem Productivity and Health under Climate Change and Disturbances. So what this is about, of course, is about biodiversity indicators, key variables that that we find in forest systems, and particularly that can provide insights into degradation owing to factors such as land use, climate change, droughts, and disturbances, and with a particular focus on bark beetles as part of a complex interacting system between climate change, forest management, and the bark beetles. And this is in regard to Norway spruce forests, which of course the bark beetles are associated with spruce in European forestry. And so the design of the pilot is an end-to-end case study, or really we're looking at a number of case studies spread around contrasting regions in Europe, and bringing together a process-driven climate ecosystem modelling approach. I'll say a few more words about that shortly, with Earth observation data, and bringing in machine learning methods as well to leverage really the complementary strengths of the process-based approach in terms of understanding underlying mechanisms and drivers and how they influence the condition of the forest and the function of the forest, as well as the interactions with disturbances such as bark beetles, and bringing that together with the temporal and spatial coverage, of course, of the Earth observation data, because the Earth observation data can always provide at best a proxy into the actual variables that are governing the dynamics of the ecosystem itself. We use the machine learning model as a sort of multivariate machinery doing its magic to bring those things together, and we think that provides additionality relative to the state of the art in Earth observation methods and products that are available currently for this sort of work. So we're focusing in terms of developing the algorithms for this on a case study in Sweden. I'm sure Rashid will be saying more about that. And we have scoped out at least a couple of other study sites in other parts of Europe where Norway spruce, either as a natural component of the vegetation or in forestry, is an important component, looking particularly at Southern Europe. So just a couple of words about the tools you may. Okay. To briefly introduce the team as well from the Lund side, and apologies, this pilot is in collaboration with ITC. Abdullah is here. I think Marga is here as well. I'm sorry for not including you. This is the Lund part of the team anyway. Myself and my colleague Tom Pugh are leading the work in the project generally, and then we have Anna-Marie Eckers-Shepherd, another senior modeler and expert on the tools that we're using. Mats Lindeskog, who's responsible for the ecosystem model simulations and the integration with remote sensing data. Fredrik Lagergren and Anna-Marie Jönsson are experts on bark beetle, developed also the bark beetle component in our model. And then we have Ali Mansouri and Rashid, who's here today, who are our AI experts. So quite a diverse team, but obviously bringing a range of skills together. Briefly, this is the model that we're using as our platform for the climate ecosystem interactions, process-based model. I'm not going to go into details there, but you can see the high-level conceptual structure of the model with its different modules. It takes in drivers such as meteorological inputs, so climate or weather, if you like, carbon dioxide concentration of the atmosphere. Important, of course, as CO2 is a component in photosynthesis. We have the phenomenon of CO2 fertilization happening as CO2 concentrations in the atmosphere go up. Nitrogen deposition, very important in Europe as well, and affecting system function, and land use and management as well, coming in as inputs to the model. It's a dynamic model. It's simulating the growth and development of vegetation at the local scale. We call that patches or neighborhoods. You can see a visualization there of a snapshot of the output from the model when applied to a particular location, where those lollipop shapes represent trees of different sizes. The colors represent different functional groups of trees interacting, competing for space and resources within those local patches. So it's an individual or certainly a cohort-based model, dynamic, simulates the evolution of a stand of vegetation in response to the drivers I mentioned. So integrated with this, we have a bark beetle model as well, and this is a sort of slide I picked out of my collection. I'd like you to focus mainly on the schematic on the left there, which explains the concept of what's going on. So this is a model that brings together the forest structure and productivity simulated by LPJ gases I just described, creating a stand structure, but also creating brooding habitat for the bark beetles, which is in the form of fallen coarse woody debris, usually on the forest floor. That's where it happens. So logs lying around and decomposing, and the bark beetles build up a population there from which they can attract living trees as well. And then storms and wind are very important because they create the disturbances that provides that brooding habitat, as can management, of course, both in terms of forestry operations leading to coarse woody debris on the site, but also the sanitary measures to remove those sites have a big impact on the build-up of the bark beetle populations and the damages they cause. So our model system integrates these things together, allowing us to look at those biotic interactions between a major pest of forestry, of course, and climate and management, and the growth of the forest interacting with that. Our work in this pilot is informed by and builds further on work that was done also in collaboration with ITC. It says unpublished there, but there's now a paper out led by Heidi and from ITC and with our team involved as well, where we, in a simple way, relatively simple way, combined the net primary production, gross primary production, so productivity metrics from the ecosystem model with highly resolved satellite data and an algorithm which I won't describe in any detail, but that allowed us to simulate spatial and temporal variation at high resolution over a study site. This one is in the Bavarian Forest National Park in southern Germany. And so what we're doing in this project here that builds upon this is going beyond just looking at GPP and NPP to looking at other variables that the ecosystem model predicts, things like the biomass of the tree, the biomass of the different species within the forest stand, the production of litter, we could look at a range of outputs. And to do this, we are developing some new machine learning powered algorithms to bring the satellite data and the ecosystem model outputs together. And as I say, applying those at initially at a study site in Sweden, and then later at additional study sites around Europe. So I think that provides the context. I hope that was helpful, and I'll throw to my colleagues then to continue the narrative.
Thank you very much, Ben. Rashid, I think Ben, he already explained all the concepts, maybe because since we don't have enough time, maybe if you could just focus on how this pilot will be linked to the three themes which we for the whole project, just just that or the novelty, I think, because Benny already explained it. It's up to you, but the time is really limited. I just want to.
Yeah,
sorry about
I
that.
will
Yeah.
try to be. I
No,
will
that's
try
really
to
good,
be
actually.
quick.
Was really good. Yeah, thanks.
Yeah,
Please go ahead.
I will try to be quick anyway. So let's wait. So I think the objective is very clear, as you have said. So we need to downscale OBSGESS outputs using remote sensing data. So bridging and learning like the facial representations, mutual ones, in order to downscale that. So the workflow, I have already presented this in previous presentation. So, but we have like data preparation, normalization, and some feature importance to select in order to catch the features that we are or the channels that we are looking for, and then model building, including different mechanisms, and then prediction and evaluation. So the data that we are using here are imported from Sweden, and also I'm testing with Biodiversity in Australia just for tests. And the OBSGESS output that I'm considering so far is PAR, NPP, LEI, GPP, and WC. The remote sensing data is in VR, NDVI, and DVI. So this is the baseline model that I used actually in the beginning to have an initial evaluation. So it's basically like unit model with attention mechanism, and also with physics guided layer that enforce some physical constraints at the end. So yeah, this is the baseline one. So I have used like different losses in order to train the model. So regression loss, of course, which is giving some numerical evaluation, but we need some physical constraints, as I've said. That's why I'm using different physical constraints, ecological ones, conservation loss, for example, smoothness, and pattern loss mainly. So the results show that the training metrics converges actually over only 10 epochs around. So the total loss decreased a lot. So conservation loss was very actually low. And pattern loss is somehow intermediate, around 1, but smoothness loss actually is somehow the hardest one actually to reduce, but we achieve acceptable, reasonable actually result on that. So this is like the total loss during the training, and for the other ones, for conservation, pattern, and smoothness. But I will explain better actually the recent work that I was doing last month. Someone is asking, or?
No, I think it was a background sound. Someone just opened it, but please go.
Okay, no problem.
Yeah, thanks.
So what I was doing actually last month, because most of what I have presented so far is done before the consortium meeting. So I was investigating to improve the model performance by including more attention-based spatial learning with some either self-attention or attention mechanisms to better capture the long-range spatial dependencies in the remote sensing data, and then also use some multi-scale feature integration. Instead of using only like pixel level, we can have something similar to pyramid network in order to catch hierarchical dependencies actually in the just all Eris data, and then to have also some loss weighting, because as I have said, some of the losses, physical losses, are easy to learn, but some are harder. So we try to have some weighting, and then some uncertainty quantification to add some probabilistic outputs to estimate how much the model is confident actually in giving such predictions. And of course, temporal consistency that I am adding in order to enhance the model to ensure consistency across like temporal sequences actually, and this will help in order to be more to catch really like the frame in a specific time. So I'm using here also attention mechanism. So I'm testing different models. I will not explain all of them, but for example, here I have TransUnit++. Not all of them are already pre-built, but I am adapting them actually to my context. We add in either some mechanisms or some layers. And I have here dual branch encoder decoder with feature alignment loss. multi-scale swing unit with physics-informed attention, and JNN, which is somehow using patches or superpixels of various data as nodes enriched with the LPGS embedding. So, and diffusion-based decoder, which is actually inspired from the normal diffusion model, which is based on the noise in effects, and contrastive pre-training with fine-tuned fusion network. So those are the results that I got so far. So the best performing actually here is I will start first by metrics. So I have around in MA I have around 0.14, 15 as error. RMS is around 0.25. So in terms of percentage, it's like 20% errors that I got so far. In terms of the other metrics, conservation is really good, as I have said in the beginning, after waiting, and I use here ratio instead of real values. So it's good, but smoothness is a little bit lower, and pattern loss is acceptable, I will say. Regarding the models, so switch unit with physics attention and diffusion decoders are like the top performing here across almost most of the criteria, and they balance this numerical accuracy with physical conservation and spatial realism, pattern, and smoothness. So then in the second rank we have trans units, plus and dual branch, and then at the end we have JNN based model, which are actually underperforms all the other models, but in terms of global consistency, it has a little bit higher effect. That's fine. Though this is your question. Next steps actually is to consider improving the used custom conservation loss for physics-informed neural networks, because I have used my own custom losses. So, but there are some libraries that I can use from there with somehow advanced, let's say, functions. But yeah, and there are also some research in the research review. I found some models that were used for downscaling different output, not LPGS output, of course, but for climate and others. For example, here they have lightweight diffusion-based super-resolution heat that I can include also in the model, in top of the model, in order to violating energy biomass sums, for example. And also downscale all LPGS variables jointly with a shared encoder. So far I am considering them as channels, but it's actually proven in this paper. It's very recent actually that joining actually the model in a shared encoder and learn the presentations independently in the beginning and then fuse them later will have like will improve actually the performance. Soft parameter sharing improved, more stable here. For example, in the case of sea surface wind downscaling, for example, in this application. Anyway, so I'm inspiring from other papers to check what will be the path to follow in order to improve the modeling. So yeah, this is the last slide. So links to other pilots. Actually, we will give actually our final models and results to pilot 5. In order to use them for disturbances, and we will collaborate also with the work package for the 5.3, and we will have actually in the next 2 weeks, when Ben will be here in Sweden, we will have a meeting actually with pilot 3.3 in order to initiate the collaboration. Yes, thank you very much.
Yeah, thank you very much. Thanks, Rashid. Although we missed one part, which is about how this pilot will be serving the 3 specific needs of the project, which is Marga shared, but maybe later on you can just add this slide into your PPT and then share it with Marga, and also maybe we can also discuss now. I think it's time to have question. If there is any questions, so 20 minutes we have time for that, for the all 3 pilots which we presented. Yeah. So if anyone has a question, please. Yeah, Andrew, please go ahead.
Yeah, thanks, Yeah, Heidi. It's actually a question for Sander and the team. We've had some discussion, Sander and team, about the how whether to and how to integrate the UNIS work you're doing into the sort of international GeoBond level, CBD level work, and specifically that's being addressed in work package 1. In other words, the norm we've been emailing about that, and some of the norms that are emerging at a global level are not, let me say, directly linked to the UNIS type approach, which is part of the EU policy. So are you planning within OBSGESSION to work on this link, and how do we do that between work package 1 and work package 5?
Yeah, good question, Andrew, because I do not have a straight answer to that. I think in let's say in the global policies, it's much more related to also the IUCN typology, and I'm not sure if there are people working also on those relationships between the UNIS and the more global typologies, because that is something that would be interesting to have a better look in how those are related on the global scale and on the European scale. Because at the European scale, there is already a close link between, let's say, the Annex 1 habitats and the UNIS habitats. And of course, it's often a many-to-many relationship, but I'm not sure what the current status is with the UNIS, with the global approaches. But I will also I think it's also, I will ask that question to Stefan Hennekens, my colleague, to see also if there are more developments going on that level, because I'm not completely aware of what is taking place there.
Yeah, I mean, I think it's important issue to sort of link the work package 5 and OBSGESSION objectives. But I know Vito, I don't know, is Elena or any of our Vito colleagues, because I know that there was some discussion in
the Wheat project
about doing this as well.
Yeah.
Are you? I mean, that's probably a place first to coordinate, right? I don't know. Are any Vito colleagues online? No.
Yeah, Ellen is there.
Yes, I'm here.
Do you have a comment on that, Eileen, or do you know? I mean, we had talked with Marcel and yourself and a few people about this, and there was some discussion. Have you got any more development or thoughts about this? I think it's certainly from a CBD and a GeoBond perspective, it's really important. And they're looking for this from Europe, some sort of leadership, I think.
No answers yet to that, but I think we need to discuss what the scope is here within OBSGESSION and whether we can do these crosswalks and make sure that it's aligned with what is happening at the global arena.
Yeah, because I see also Elnaz is there. I don't know, Elnaz, you were more up to. I mean, we haven't had a meeting. We had some meetings about a couple of months ago on this, or 3 months ago, and we'd sort of left it. And I'm a bit afraid if we don't take action, it's going to just sort of drift into the background and nothing happens, And it's just a pity to me that there is such a clear, as you said, Sander, push to coordinate with the IUCN typologies, and Europe seems to be going in a very different direction with this, and it just means sort of leadership in biodiversity is going to be lost at a policy level, and that then comes back into the OBSGESSION kind of these key 3 objectives, which need to be addressed. I think I don't know.
I agree with you, Andrew, but let's say to make good crosswalks, that is something really we need to have to the ecologist in place because sometimes this is complicated. Sometimes it's very easy, but you have to know, you have to involve the ecologists. So I will discuss that also with our ecologists to see what is taking place there, or we can take specific actions on that.
Yeah. I just, Elnaz, do you have any comments on this at all or any updates?
Yeah, sorry, but I just came out of another meeting, so I'm not sure that what is going on, but I grasped some points from your talk. I think in work package 1, we try to address this issue because, as you said, is it very necessary to pursue this path to work with the CBD and also connect the EBVs with the CBD's requirement, particularly when it comes to ecosystem extensions. But I'm not sure what was the exact discussion here. Sorry, I just came out. But this is what we address in the work package 1.2 of the obsession.
Yeah,
and
I guess the question around us was whether how to make that link between the policy work in work package 1 and the actual work in work package 5, and particularly, the UNIS type approaches which are being pursued in Europe with the work package, with the ideas coming out of CBD and Geobon more around using the IUCN typologies.
I think this is the extra work that we decided among partners in work package 1.2 to voluntarily address that, to how we can do this link. This is not in the proposal, but our partners in work package 1.2, we are waiting to see that how we can connect between the work package 5, that is a biodiversity, with our policies in task 1.2. This is for sure is back of our mind, but it's working on progress, and we have come up with the reviewing of the current or existing network and document about the policies and also knowledge gaps in this particular case. But we will, it is in our agenda. I make sure that we will tackle this one because it's very important.
Yeah, because that's still not. It's partly related to the UNIS level 2.
Yeah.
Ecosystem. So in the end, you would like to see that also at level 3, I think. But at least that's a good start.
Yeah.
Thank you. Is there any other question? Okay, then maybe I do have a question oh yeah, Ellen, please go ahead.
Yeah, I do not have a question, but a request from work package 2. So maybe Heidi, if you have a question related to the pilot, then you can go first, and I will,
Okay.
because it's a more general request to all the pilots, not a specific pilot.
But you can go ahead because mine is just related to pilot 2. So yeah, please go ahead with your question. Yeah.
Okay, yeah. So a message from work package 2 that we will soon start benchmarking a couple or selection of the products that are described in Andrew's paper from 2021. There's a lot of products there, and probably we will not benchmark them all. So therefore, we would like to know from the pilots which products are most relevant for your pilot, and then let us know. I know Marga has sent an email around, but I think this is for us quite crucial to know this. So please respond to that. Check the list. Respond to that email. Otherwise, we will have to make a pre-selection ourselves, and then maybe it's not the most relevant products for your pilot. And I think the pilot responsibles or people involved are most are the best ones to select or to indicate which products are most relevant. And then a second request is we presented the data catalog, the Earth observation and in situ data catalog at the annual consortium meeting. And the question is whether, or a request is, please fill those with data sets that you already have produced, because we saw little activity there, although Tijana and Susanna asked to fill this data catalog. So it would be good if you could dedicate some time to that, so that we can start preparing also deliverables based on the data sets that are in there. Yes. And then I saw that Sandra has a question. Is that to me or?
Yeah, that's to you, because we responded to the request of Marga, but that was, let's say, more related to a wish list from the pilots before, and that's not one-to-one to the list of the EBVs from Andrew. So maybe we're not working completely with the same list, because the benchmarking you said is on the, let's say, on the list of the EBVs, as in the paper of Andrew and co-authors, while in the spreadsheet is much more on specific remote sensing products. So there's some overlap, but maybe it would be nice to have an overview of what are the potential, let's say, EBVs you would like to work on, and then see how this relate to the Excel sheet.
I think Elena is frozen, but
Yeah, I think so.
yeah.
Okay.
Yeah, I don't know what
too
she-
afraid for the
Yeah,
question.
but maybe Sandra, you may need to repeat when she back actually, because I don't know if she already heard what you said. But yeah, but let's wait until she will be back. But then I will have a question if Rashid, if he's available. Rashid?
Yes.
Yeah,
I'm
thanks.
available.
I do have a question, because I saw from one of your slide, you mentioned that yeah, you are also going to downscale the LPG gas product. We are also interested in our pilot 4 to know which product you are going to downscale from LPG gas model, because we may also use those in our pilot to assess biodiversity loss using the eDNA data. So if you could just explain a bit, or at this stage you.
Yes, actually, our aim actually is to provide a generalizable method somehow, in order to enable all the to downscale all the variables. But so far, I am focusing more on FBAR, NPP, and LEI, GPP, and WC. So, but if you are interested in specific variables, then we can work on them, prioritize them better.
Okay.
Yeah, because our aim actually is to make something general. And regarding your question in the consortium meeting, so I think you have published one paper for downscaling for LEI.
Yeah.
So I think because somehow, you have used the OLS, linear regression. That is considering actually that the relationship is linear. But for most of these actually variables, and particularly if you try to use not only one as input, but many, so the relationship is nonlinear, and it will be harder actually to catch using simple models.
Yeah.
And also for LEI with the EVI that you are using, so the relationship will become nonlinear after around 6 meter square over meter square. So yeah, the hypothesis or the assumption of linearity is not actually holding everything.
Yeah, thanks. No, I think the paper which I mentioned during the annual meeting was also, I think, one of your colleagues involved. I think Ben also mentioned in his presentation. So like the product from the LPG gases came from his work, Mats. So he was, let's say, heavily working on the product, and we just, let's say, evaluate the result based on the impact on bark beetle, how bark beetle affect the MPP changes at high resolution, special resolution. Yeah, but thanks for the explanation. I think it's, yeah, maybe we can arrange a meeting, because I think you also need some input from our side for the disturbance data. Yes, so we can arrange it, and then we can see how we can cooperate in more detail.
Okay,
yeah.
thanks.
Sorry, any other further questions about the 3 presented pilots? If not, then I think we can take a 10-minute break.
Thank you all for coming back for this presentation after your coffee break. So yeah, this is a very short presentation of Pilot 4 about ecosystem condition of temperate and boreal forest protection and restoration from soil eDNA combined with image spectroscopy. The general objectives of this pilot is to use a combination of remote sensing data and environmental data from soil samples to evaluate the impact of forest disturbances and environmental stress on biodiversity. And this using both environmental eDNA-based indicators and remote sensing derived products. The innovation of this pilot is the integration of these two types of information, remote sensing information and soil eDNA, to unlock new insights in forest ecosystem monitoring, and use that to develop a scalable framework to be able to monitor ecosystem health and biodiversity shifts and changes linked to changes in forest disturbances and or stressors. So the expected impact of the pilot is that we will be able to conduct fine-grain monitoring of biodiversity changes from space by coupling next-generation satellite remote sensing and eDNA profiling. And by combining those two very powerful state-of-the-art techniques, we'll be able to monitor change of biodiversity for the first time at a continental level and in a very consistent manner, because we use two standardized methods. And that will allow us to create and monitor the environment using fine-scale continuous resolution maps, and look at a very detailed level that the environmental DNA can give us, more detailed than you can get from normal point-based human field observations, and that will deepen our understanding of how biodiversity is impacted by both anthropogenic pressures and natural impacts. So this all links well to the main mission of OBSGESSION. So we will monitor and predict both biodiversity change and its drivers in terrestrial ecosystems. For this, we use an integration of state-of-the-art Earth observation data and in situ environmental DNA data, and use next-generation statistical models to make ecological predictions. One aspect we're not very sure about how to link it is to account for uncertainty in our models, and we will need to find some extra collaborations with Work Package 4 to see what are the best ways to incorporate uncertainty in our models. Because of a lot of the uncertainty, for example, in the eDNA data is not fully known. So it would mainly be the statistical uncertainty of the models itself that we can incorporate and account for. If you look at the specific needs for biodiversity, we contribute with this pilot to harmonize biodiversity monitoring by understanding and evaluating forest soil biodiversity, especially microbial biodiversity in forest soils, and use standardized methods, protocols to create pipelines that will produce consistent, comparable, and scalable biodiversity products that will allow large-scale and harmonized biodiversity monitoring. To do this, we will make an improved use of Earth observation data by combining it with state-of-the-art soil eDNA data to enhance biodiversity monitoring and thereby providing a scalable approach to detect changes in soil community composition caused by forest disturbance and environmental stressors. This will allow us to investigate drivers of ecosystem change, to do this on a scalable level, both on the location of pilot sites, but also European level, and produce base maps based on these two techniques that will allow us to study changes in forest ecosystems. These methods are based on proof of concept methods that we developed during the previous Biospace project, where we used a combination of hyperspectral data and eDNA to train machine learning models to produce baseline maps of microbial communities, both patterns in diversity or relative abundances of specific microbial groups. So in OBSGESSION, we are building on these methods, improving them, expanding them, and linking them to forest disturbance events and natural stressors. So for that, we use both local scale data that we have from our previous project, but also EU scale Lucas soil biodiversity data from 2018. There has been a Lucas soil biodiversity collection event, I think in 2023, if I'm correct, but we're not sure whether that new data set will become available before the end of the project. We combine this with mainly multispectral and hyperspectral remote sensing data to build our machine learning models to create maps of microbe diversity on local and continental scales, and we will then use additional layers of environmental variables to either test whether ecological patterns we see in our data make sense, or to test on the effect of forest disturbance stress variables on microbial communities. One step that we're already doing in this is to understand which part of the forest soil microbiome would be of interest to model and study, because the soil microbiome is extremely diverse with a lot of different groups being involved there, making it possible to create models for all those groups. Just have a paper under review where we're highlighting that in forest, bacterial communities are a good proxy for changes in biodiversity of the other soil groups, because the bacterial communities correlate very well, especially in terms of beta diversity, with the beta diversity of almost all other soil groups, and do this consistently over different forest types. This is based on one of our data sets we have of the Bavaria pilot site. And based on that information, the first models we are running on European scale are based on bacterial diversity, where we're now looking at especially which aspects of diversity, which biodiversity indicators we should use, and how scalable our results are. And this is still work in progress, and in later steps we will correlate these maps with other data layers to understand the ecological patterns that we're seeing. And that's at the moment the state of our research where we are. Heidi, is there anything you would like to add to this?
Thank you very much, Angie. Nothing no, based on, so far the idea of this, let's say, meeting today is just to show how we link our pilots with the 3 needs from the whole project, and the novelty of which is, I think you already presented very well. Thank you very much, Anjan. I think we should. Thank you. Thank you too. We should forward to the next pilot 5, freshwater, and then later on we do have time to have a question about if anyone have a question about our pilot or the other pilots which is presented during the session.
Okay, perfect. So moving on from terrestrial to freshwater, pilot 5, monitoring freshwater ecosystem functioning under climate change and disturbances at European scale. This is a collaboration with colleagues from Brockman Geomatics and Süke. Yes, we have three main goals within this pilot. One is the data production with a focus on freshwater ecosystem functioning. Especially here, we focus on phenology and disturbances, especially harmful algae blooms. The second one is target change and attribution of change in selected lakes, and then also to assess the potential for upscaling to the European scale. And we already identified collaborations, which is already ongoing and also mentioned with Pilot 6, so the other freshwater pilot. And I think the collaboration in the future, which will start also with work package 3, especially here for the detection and the attribution of change, so our goal 2. We addressed each of our goals to the specific needs of OBSGESSION in the next slide. So our first goal, as mentioned, the data production. We here focus on phenology and disturbances, especially harmful algae blooms in the first place. This can be linked to the harmonized biodiversity monitoring from OBSGESSION. We focus on these both remote sensing enabled variables because we have their algorithms in place, which we can process and then describe the ecosystem with. And we did this, or currently doing this, for our pilot study sites. They are known. I will go into more depth later here. To go a bit more into detail to both of these variables. So the first one is the freshwater phenology. This is especially addressed for bloom detections and especially shifts in phenology. We developed an algorithm in the last month to detect freshwater phenology. So there's currently no operational service available, not from Copernicus or CCI. We focus on Sentinel-3 Olchi. So this is an water quality remote sensing sensor. And we focus on the chlorophyll concentration because we have here a temporal or better overpass from the satellite. So the satellite often comes every day or twice a day over our pilot site studies. We also can apply it to Sentinel-2 or emerging of both of these sensors, but using only Sentinel-2 is a bit more complicated because the repetition rate is not as high as for Sentinel-3. Here you can see two images, so I will start with the lower one. Sorry. There you can see 3 years, and this is the chlorophyll A concentration of the lake is displayed, and then you can see the freshwater phenology detected by the algorithms. So in 2022, we have 3 blooms detected. We have here a bloom in spring, so the spring bloom is very dominant in freshwater ecosystem often. We have then a smaller bloom detected and then a larger summer bloom. In 23, we have also a quite small spring bloom and then a larger summer bloom. And then in 2024, we have only 1 bloom detected, but you can see the chlorophyll concentration is quite stable on 1 level. So that makes it a bit more difficult for this freshwater phenology detection. Freshwater ecosystem can be very diverse and depend really on the meteorological and weather phenomena. So do we have enough sunlight? Do we have the temperature to the phytoplankton to bloom? That's why this detection of the phenology is quite important to describe the ecosystem. And then on the upper map, you can see this is Lake Mellerin, one of our pilot sites. And here we can see the start of the first bloom divided into different water bodies. So we can not only describe the phenology for the full lake, but also go into sub-basin level, where we can then see different phenologies for the water body, so for the sub-water body parts. And you can see the darker the color is, the later the bloom. So we have for this, for example, very large water body part is the bloom quite late in 2024. And for the smaller water body parts, the bloom starts earlier. Yeah, we can also include the chlorophyll concentrations from services. So we already have services in place where we can get the chlorophyll concentration from, which is Lake CCI and CLMS, and we are thinking about including them once we go to the European scale. For the second variable, this is the cyanodetection with Earth observations. There are various publications and algorithms addressing this, especially immersing cyanobacteria, so in the water column, and also floating cyanobacteria. So cyanobacteria can go float on top of the water body, and then it's a very dense biomass, yeah, mostly like floating vegetation even. So this can also be detected. The different algorithms can or detect phycocyanin mainly, and this can be cell counts or just abundances. And the algorithms are often based on the water color sensors, so especially Sentinel-3 OLCI and MERS, because they have a specific band in the absorption feature, which you can see here on the right. These are different absorption features of different pigments, so chlorophyll A, which are the green ones, and then the cyanobacteria one is the blue one, and the red one, so phycoerythrin and phycocyanin. And we are mainly focusing on phycocyanin, so this is the blue absorption feature, which is mainly present in cyanobacteria. The issue with the water color sensors is that the spatial resolution is often not enough for small inland water bodies. So Sentinel-3 OLCI is 300 meters by 300 meters. So if we want to go into smaller water bodies or sub-basins, it's often not enough, or we have less information. And therefore, Sentinel-2 MSI proved to be a valuable additional sensor. The issue is that Sentinel-2 doesn't have a specific band on this absorption feature. As you can see here, the gray bars are the Sentinel-2 wavelength. So we have B2, B3. Then we have a gap just where the absorption feature is most dominant of phycocyanin, and then B4 and B5. So although this sensor is not made for cyanobacteria detection, we have an algorithm in place which makes use of machine learning and which gives us a specific risk of cyanobacteria presence. So it makes use of very strong cyanobacteria blooms which can be detected with Sentinel-2, but with a higher uncertainty. And here you can see some results from Lake Mellerin, so one of our case studies. On the left side, you can see the OLCI image. If it's red, then cyanobacteria are detected. This is one of the sub-basins where cyanobacteria often occur. And on the right side, you can see this it's a similar period. Sentinel-2, you can see that the resolution is way better. And here we have also a risk with also in the SPAY having, especially here, very high certainty of cyanobacteria presence. I think here I put an RGB image. It's maybe not so good to see, but often you can see already in the RGB image, if we have a very strong green, light green color, this often indicates already that cyanobacteria may be present. And in the last month, we intensified our validation practice. So Petra took a closer look into the validation of cyanobacteria for Lake Mellerin. We therefore had some in situ data from Sweden, and we compared it to our algorithms. And here you can see we had 24 matches in total. So there was a in situ cyanobacteria measurement where we also had a valid overpass from Sentinel-3. And you can see that the accuracy is quite good. So 12 times we had also cyanobacteria presence detected from EO, which was also detected from in situ, and also the negative, so no cyanobacteria from EO and from in situ. We had 2 mismatches where our Earth observation algorithm said that there are cyanobacteria present. But we already identified that this once was, I think, very dense pollen from the trees, so that was a very green color, which where the absorption spectra was quite similar to cyanobacteria. And the other one was a very hazy day, so this was a flagging issue of Sentinel-3. So we could also already explain and identify here why we detected falsely cyanobacteria. The benchmarking is currently ongoing. So the two products we showed will be benchmarked for the products, and for the algorithms we already started to select the algorithms we have, and we'll also go more into details into literature research if we have other algorithms which we want to validate. Especially for cyanobacteria. For phenology, it's a bit more difficult. Although there are multiple approaches available, the methodology to derive freshwater phenology is often quite similar, and the validation is difficult due to temporal limitation in situ data. So the in situ data are often just measured multiple times a year, so that a freshwater phenology cannot really be derived from in situ data. So we only can rely here on really sensors in the water, which are not that common. I think the pilot sites are already known. So this is Lake Mellerin, Markermeer, and Kökköminjoki, which from pilot 6. I think I don't need to go into detail here. That doesn't it didn't change a lot. We'll skip this. Going to goal 2, the target change and attribution of change. So this is linked to the OBSGESSION specific need knowledge of drivers of ecosystem change. Here we want to strengthening the understanding of freshwater ecosystem and their changes, because we have quite good time series of EO-based remote sensing enabled biodiversity variables. I think we can go more into detail here. And we know that there is auxiliary data available, especially for drivers. So there's meteorological data which we can access, land cover data, and also other. So here we really hope of some guidance from work package 3. And it's more than welcome. I saw already that there's this evaluation ongoing, and I had a quick look. And we identified 2 methods from work package 3, which we think are very interesting. So one is the independent detection, so especially the change detection and identifying trends, especially for the cyanobacteria part. This is very interesting. And then the causal discovery and causal machine learning, so to better understand causal structures here in freshwater ecosystems. And yeah, here's just an example again from Lake Mellerin for the cyanobacteria risk. And you can see we have the full year from Sentinel-3 Olchi. 2025 is now processing. So from 16 to 2024, and this is weekly aggregated. And here we have a cyanobacteria count, and if it's very dark red, we have a very strong cyanobacteria bloom. So often cyanobacteria are often detected, and you can see that we have some trends. So we have a trend towards also cyanobacteria in the spring, and also maybe denser or more often occurring cyanobacteria also in the summer. So here we want to dig deeper with work package 3. To investigate these changes in time and space. So how do we best detect the trends or even shifts? And can we identify here drivers? And our goal 3 is then the upscaling to the European scale. And this links to the OBSGESSION need improved use of Earth observation data. So once we have the benchmarked algorithm used and processed, we can also assess how we can do this on a European scale. We have the option to use already services in place, so either CCI and Copernicus data, but these are only for selected lakes, I think around 600 in Europe. So we need to see if we want to process all the water bodies in Europe or happy with the ones which are provided by the services. And then also potentially apply the detection future of change models to the European scale. So once we identified with work package 3 a good model, if this can then also be applied on the European scale. Yeah, and to the timeline. So just presented these 3 goals and the linkage to the OBSGESSION specific needs. For goal 1, so the processing of the data is ongoing. For the pilot sites, validation of the pilot sites is also ongoing. So Jenny from Suke will provide us with some validation data also for Kirkcaldy, and then the validation will be done there, and the benchmarking is ongoing. For goal 2, we just we are hoping for support from work package 3. We just saw that they are evaluating the different evaluation techniques and approaches. We are really looking forward to this one. And we saw that there are all these different criteria we can also apply with our data. So there the interaction hopefully will be starting after the method assessment is done by work package 3. And goal 3, so upscaling to European scale will start once we finished goal 1 and goal 2. Yes, that's it from work package from pilot 5.
Thank you very much, Joris, for a detailed presentation. So now we look forward for the last pilot.
So this last pilot is the pilot number 5.6, ecosystem functioning and freshwater and transitional waters quality. This is the original name, but we have also ongoing work related to the terrestrial side. So I guess that we have a bit changed our focus. But yes, our pilot is working on a catchment scale, and Jorit was just referring to it as it is a Kokemäenjoki pilot, Kokemäenjoki drainage spacing, and also its estuary. And we are working on ecosystem functioning, freshwater quality, terrestrial land use and phenology analysis. And our field work is focusing at the moment a lot with eDNA work, and I have some specific slides for that, but also water quality and freshwater phenology, as Jorit was just explaining. And over the area, we have quite well-established linkages with citizen observations, and these are especially related to algae bloom detection during the summertime. And our Earth observation data set, I will explain in detail in further, but this also links to work package 2, but also we have some national Earth observation data sets that we plan to use, and also so correspondences between the work that is done in a European level in OBSGESSION. And then the EPVs, both freshwater and terrestrial, and the dam modeling are linking our work to of course, to the other work packages. And there, especially the habitat mapping and combining Earth observation data with some detailed in situ of structural habitat features. This is the linkage between especially work package 3. And I won't go in details with our pilot area, as I guess that we have already introduced it quite many times and referred also today. But the linkages over here, which I'm referring to, are mostly related to the work package 1 and the biopolicy perspectives that we have. Of course, there is the biodiversity strategy, but also with the work task 5.5 and 5.6, we have the well-established links to the data requirements in Water Framework Directive in specific, and then the biodiversity strategy and the recent restoration law, of course. And at Syke, within our pilot area, we have an ongoing service related to water quality. This is Syke's Open Earth Observation Service Tarkka that provides automated access to water quality within the lakes and also in coastal and open sea areas of the Baltic Sea. And within our catchment area, we provide information over the lake side for growth rate, turbidity, and temperature. And within OBSGESSION, we will complement this tool by combining Earth observation and also citizen observations for the final themes of interest. And this is to be decided, but as mentioned, some of the freshwater water quality parameters are already provided, but especially the work related to phenology and species information, this is the development work we want to put forward during this project. And so just a summary on the data sets that we are working on. So within freshwater, we will work with Work Package 2 provided datasets and the linkages with Pilot 5.5. And Jorit was already explaining this in detail, so I won't repeat them. But chlorofluoride is the main freshwater phenology parameter that has been chosen. And also there is work related to algae species and biomass information and the continuation with Syke's algae bloom work that inherits from previous projects. And I already mentioned the National Earth Observation data. So we have a National Earth Observation database on water quality information that provides us with the national chlorofluoride interpretation, turbidity, surface temperature, and also Secchi depth information and CDOM, but we are not working on these during this project. And on the terrestrial side, there is the dataset that we will get from Work Package 2 and related to terrestrial side. But also we have the National High Resolution Marine Land Cover Harmonized Time Series, and then with some very high resolution land cover with vegetation height, and this is from the year 2022. And changes in vegetation structure from airborne LiDAR campaigns to national campaigns. The other one is, well, you can see the timeline over there, but they are over 5-year periods, both of them. And then there is the multi-source forest inventory data, forest parameters from 2009 every 2 years. And then we also have the harmonized Landsat time series starting from 1984. So these are the national data sets we are working on, and also there is inherits from previous projects. But then the field data sets, and especially related to this project, the eDNA sampling was done already during last year, and I will show you in a minute the plans that we have for this year. And also we decided to save some sampling to year 2026 to cover all of the years, all of the relevant years during the project, and also to be able to analyze the previous samples before making the very last field data sampling. And then in addition to these specific campaigns, we have lake monitoring stations from environmental administrations, continuous measurements of water quality parameters and algae species over certain stations. And since we have the pilot area is quite large, this is quite a large data set of water quality information. And in addition, we also have automated river station measurements within some of the rivers that link turbidity and nutrients quite nicely to the Earth observation data also. And I mentioned the citizen science networks, which is related to algae blooms already. And so this was already explained during the presentation of Pilot 5.2, so I won't go in details, but there is a collaboration going on, not related to the Kokemäenjoki drainage basin, but especially the areas in the very northernmost part of Finland in the Lapland. And as this was already dealt, I will just comment that this is related to the pan-European UNIS habitat type suitability maps. And so this is linking the previous work done in Finland and very detailed work related to the habitats in Lapland to the more European specific habitat mapping. And I hope that there will be some nice progress in this. As it was already mentioned, it was a couple of weeks when the first meeting was done, so we look forward to the conclusions during this year. So to the eDNA sampling part, this is a previous slide that you have already seen about the fieldworks done during autumn 2024. And there was eDNA water quality analysis, stream organic matter experiments with cotton strips. And I guess Tina is not anymore present in the meeting, but there is Henna in case you have some specific questions on this afterwards. And there was already a published paper related to this and eDNA work development in Finland by Tiina Laamanen, Technology Readiness Level of Biodiversity Monitoring with Molecular Methods. So I hope you familiarize with this yourselves with this in case the topic is relevant or interesting for you. And for this year, the planned fieldwork is focusing on especially for lakes. And we focus on lake eDNA sampling and water quality. And this will be done in August during this year. And we have selected some specific lakes that are of interest to freshwater topics of these projects. And these have been selected together with Pilot 5.5 so that we can link this also to the European goals of this, European level goals of this project. And I will show some details in the forthcoming slides, but also would like to mention that there is work in progress on combining the existing soil and leaf eDNA work with remote sensing data from Evo area, which is in the map over here. And there is an article preparation in collaboration with the OBSGESSION partners from University of Twente and Syke, and perhaps with some other partners also by Topi Tanhuanpää is preparing this article. So yes, to go back to the freshwater side, these are the lakes that have been chosen for eDNA sampling, and they all are interesting and relevant also from the algae species means and with the phenology part. And there is a nice time series of water quality measurements within these lakes, and especially in Lake Vesijärvi also some automated stations where we can combine with the flow-through data. And to mention with this eDNA sampling and freshwater development, there is also, we will be receiving a complementary data set from Finnish Uvaspace company that has a hyperspectral instrument. And we will be receiving during this year about from 10 to 20 observations of hyperspectral satellite instrument with resolution of 25 meters. So this will be really interesting experiment and supports nicely the goals of OBSGESSION, especially for making preparations for future satellite instrument development and work. And as Dorit was just explaining, that with the current Copernicus datasets, with high resolution needs that we have over the freshwater side, this instrument is really suitable as it has a nice spectral range and capabilities of observing also the algae species information. So we don't have experiences on the data yet, but after the summer, we expect to have some interesting results. And this is an example of time series of cyanobacteria bloom over the lake Kirkkojärvi within our catchment area. And over this lake, there is practically every year's very dense cyanobacteria bloom phenomena. And so this is one of the lakes where we will have also the eDNA and water quality sampling. And as Dorit was explaining, the main goals of the freshwater in European level within OBSGESSION, we are focusing on chlorophyll and phytoplankton biomass information also over the Finnish lakes areas, and we have been collecting the field data set for validation for Dorit over all of the lakes within the catchment area, but especially focusing on our pilot lakes. And this is just a quick example of our Earth observation service providing information on water quality. And this is Lake Vanajavesi, which is one of our pilot lakes, where we will have the DNA sampling also during this year. And above there is an example of the total biomass information time series that we have over this lake, and the species information. I won't go in details with this today, as I think that the last pilot doesn't need so many details anymore. But anyway, this information is what we have available over quite many of the lakes within our catchment area. This is a map showing the most relevant lakes in red, where we have the phytoplankton data sets available over the drainage basin area. And also we have been calculating on the number of samples to identify the main lakes where we really can focus on the validation information. Sorry, yeah, there is some calculation of total biomass, but I guess that I can skip this quite detailed information. And over the Evo area, as mentioned, there has been a long-term development both in eDNA and terrestrial Earth observation data, and also some previous hyperspectral data sets available. So we will also focus with freshwater development over there, and Lake Pääjärvi is the main lake where there is a lot of water quality and biomass information available. So this is just an example of the data set over that area, and this is, I guess, the final slide on the development. This is linking to what we promised during the proposal, and also to link between the terrestrial and freshwater side. Also, just to mention that we have some automated river data, especially on turbidity and nutrients that are available during this project, and these are related especially to turbidity and total phosphorus, where the linkages between the automated data and Earth observation data are quite clear. And I guess I won't go in too much detail with this, but this is just demonstrating the linkages from between the work package 3 and our pilot, and also with pilot 5.5. I think this was quite joint between the two freshwater pilots with dam modeling. And there is a plan to experiment with the dam model, both over the terrestrial side with some bird and butterfly communities, and then with the aquatic dam models with the lake phytoplankton communities. And this work hasn't really started yet within our pilot areas, but I think this is really motivating goal to proceed further during this year already, I hope, but I think we plan to do work related to this, especially during next year. Okay, I think this was already discussed quite a lot during AGM meeting, but just to show you with the linkages that we have defined with our pilot and with other work packages. And the list with the linkages with other pilots, I think there is quite nice collaboration with most of them. And sorry if I missed linkages between pilot 5.3, but the linkages are really active with especially 5.2, 5.4, and 5.5 at the moment. And with hyperspectral data sets after this summer, once we have the collaboration campaigns with Kuvaspace, hyperspectral Earth observation data, and we have some more experience on their data sets, then I think we could activate more with pilot 5.1 and hyperspectral data sets linkages. Okay.
Yeah. Oh, sorry.
No, this was just, I guess, that I'm repeating if I go through this, but I just made an overview on the. We are at the moment working a lot with improved use of Earth observation data, both over freshwater and terrestrial side, and with both Copernicus dataset and of course with historical Landsat datasets. But our main focus is on hyperspectral Earth observation dataset and preparations for the forthcoming ISA-SIME, and where we can see the benefits of hyperspectral satellite observations in biodiversity monitoring.
Okay, thank you very much. Yeah, so I think we do have around 30 minutes for a question. I think maybe we can also consider all the pilots together. So yeah, now if you have any question, we can take the questions, but maybe I can start myself. I just have a quick question regarding your image spectroscopy data, which you mentioned in your pilot 6, Kuvaspace. If I'm not wrong, is it the private company? Am I right?
Yeah.
So, I mean, how will be the possibility to get this data for the other places, or is only for the Finland? Because I know this company is existing for a couple of years. I know, and it's a patent based.
Yeah, I have been actually, I tried to ask also during this week to get a response on this, because we had a discussion about this topic already in our meeting a bit more than a month ago, but I didn't get a clear answer. I think that we will need to experiment during this year, and then when we see how beneficial the use of the data is, then we can discuss more. But we have an NDA, and we will we will for sure be able to present you with the results within our Greenwich Basin area. Yeah,
thank you.
Just to mention that they are a Copernicus collaborative program company, so there should be availability with the data in the future. And I think that their next satellite will be launched together in collaboration with ESA at the beginning of next year. So in that sense, the data availability seems promising also for research purposes in the forthcoming years.
Yeah, thanks. Any other questions regarding the other pilots?
Right. Should be putting my hand up.
Yeah, it's okay. It's okay. Yeah, please.
Yeah. With pilot 4, I think there's obviously some thematic overlap between our pilot 3 and pilot 4. It's really interesting work. I was trying to understand though whether your end goal, the end goal of your algorithm is to quantify these biodiversity metrics for the soil community, or is it just should one be thinking of the soil biodiversity metrics, the alpha diversity, etc., as being a proxy for broader features of the ecosystem condition or health or biodiversity? Because as far as I understood this I may sort of misunderstand the goals, but I thought this was about understanding generally the condition of different kinds of forests through the lens of the eDNA. Probably just my naive understanding, though.
So thank you for your question, and I hope that I can try to explain it a little bit better. So there are two main goals. One is to create base maps on European scale and local level of soil biodiversity, at this moment mainly bacterial biodiversity. And then the second one is to use those base maps to explore the effects of disturbance effects or forest stress health effects on soil biodiversity at larger scales.
So the
right, that's what
that, I think, Ben, your question, how much, I think you're leading to the idea of how much is soil biodiversity actually linked to, let me say, macroecological biodiversity, trees or animals,
in a causal way,
whatever.
Exactly.
Now, that, of course, very open question, ecology. How much are any of these indicators proxies at very different sort of scales and levels? So, yeah, I mean, Anjan's second point kind of gets at that, right? That you can sort of see that there are links, correlations, if you will. Exactly what those are, totally understudied, I would say, in environmental sciences. You're dealing with different worlds, huh? I mean, microbiome, you get a square meter, you've probably got as much biodiversity activity as you do on for other highly studied sort of macroorganisms at a global scale. So it's kind of I think a very open question. And it's not to sort of say one is more important than the other. And I think it'll start to come out of this sort of work for the first time. We can start to get data at these different scales, which allow you to understand it. So that's just a bit of an infill. I don't know. I could understand where you're going with it. If I'm interpreting your question correctly.
That does reconcile my understanding with what I've heard presented. Well, and then just to say that I think there's an opportunity as we develop our products in the two pilots to look at opportunities of comparing, contrasting, correlating. If we're applying the methods to the same systems, if that's sort of within scope and the resources we have, that could help to have at least a first sniff at that sort of question around causality in both directions, or at least understanding those links at a little bit deeper level. between the, as you say, the soil microbiome versus the larger scale ecosystem that the soils are supporting or part of. Yeah. Thank you.
Any further questions?
I had a question for you, Heidi, as chair.
Yeah.
I mean, not exactly here, I guess, because Margaret called this meeting, but you're very ably and wonderfully chairing here.
Thanks.
What do you think? You know, the broad idea or the way Margaret was thinking and yourself about calling this meeting and trying to explore how does these projects better fit or do fit into the overall goals of the OBSGESSION project. What's your take on this and other colleagues? I mean, are we getting good explanation? Are we getting a good linkage back into these broader goals, or is this still something that needs more work? And do we have to perhaps call another meeting to try to get people more on the same line? I mean, I'm a little bit myself, some are really covering this very well. Others thinking, well, not quite sure how this fits in yet, and maybe we need to have more thinking, more discussion as an overall Work Package 5 team on how actually to get there in terms of the linkage with OBSGESSION, the main goals of OBSGESSION.
Exactly. Thanks, Sandro. I think that was the main goal of today's meeting, actually. And as you noticed that we all presented how we fit to those goals, but I think we need further meeting and also to look over the presentations, which is because after this meeting, I mean, all the pilots will submit their presentations. So maybe we can harmonize them to make a kind of the overall aim and for each pilot and how it fits to the overall goal of the whole project. But yeah, I do agree. And I think the session is recorded as well. So we can also take out all the key points from this perspective, actually.
Would it be fair to say that the first one is the one that people find most troubling, the harmonizing?
Yes.
That I must admit I have trouble even understanding that concept. I mean, we're developing a bunch of different algorithms using different data sets and bringing different tools together, I guess. So what would be an example of how that feeds into harmonization of biodiversity monitoring? That's the question I'm struggling with anyway.
Yeah. Any explanation, any added to that question?
I'm just brainstorming, right? I'm sort of bouncing back to Ben. Is this more to how it links more broadly into sort of like policy objectives and, is that the term harmonization? Because if you look at the full range of the OBSGESSION objectives and aims and goals, it seems to me to be largely how do you get products back, actually into useful for either the economic sector, that could be industry and people doing, environmental impact assessments and all that sort of business. But it also is very important, of course, for policy level, at all, from local to global. Is that what we mean by harmonization, and how are we linking back, or is it harmonization across scales? Is it harmonization across, trophic levels? I guess it's open was the way I'm looking at it. And does that need more focus? So I think it's a good question, Ben. Perhaps that's something that, could be better explored by the group, and how to put this together. Maybe each of the work package leads should get into a huddle, the task leads into a huddle and try to start nutting out a few kind of broader concepts on this. That's my sort of observation as a having listened to these very interesting and they're all really great talks and really great tasks, and sub-projects people are doing in this work package. And so how to bring it together into a coherent whole, I think would be a great step forward, actually. So I think it's not, I think it can be done. It just needs a bit of work and focus on the group, actually, and particularly work, each task lead perhaps to get together with their colleagues and then try to build this up into a bigger picture. Yeah. But it would be a really, really cool, contribution to science as well, I reckon, if this is done a little bit more, a bit more thought and a bit more care. Really interesting. Yeah.
Thanks, Andrew. I think, Engin, you also raised your hand if you want to add.
I'm a pretty visual person, so what I maybe miss is something like a table or a network or something in which we can connect the concepts and the projects together, instead of just presentations and text on a presentation, but more a project that can maybe taken into the reports, etc., to show the interconnectivity. Could be some keywords coming out of the aims and the specific needs of the project, and just links to the different pilots with arrows and some keywords. But I think we need to go beyond just presenting what we do to each other to creating together one simple products that in one view you can see the connectivity. That would be a little bit my intake here.
Thanks, Engin. I think we do have such a type of overview, but I don't know to what extent it's, let's say, developed or it's in a final phase. But I'm aware of that. Like last year, there was a couple of emails exchanging to ask each work package to explain and then develop such a kind of flow, how these work packages link together, and also what they provide to the overall goal of the project. But maybe we can follow up on that, actually. Yeah, I agree. Thanks. Yeah. Any further question, comment? If not, then we can close up the session. And yeah, please, if you still didn't submit your presentation PPT, please submit it to the folder which is Marga shared. Yeah, and also the session is recorded, so you can have it after I think a couple of minutes after the meeting on the chat. Yeah.